So we take for granted that we have unspoiled food in the fridge, drinkable water that comes out of our taps, and shelves full of antimicrobial products. In fact, in our lives in this country, the degree to which we control microbes is a monumental concern in our lives. So in this chapter, we'll be dealing with some of the terminologies and mechanisms that we use for physical and chemical control of microbes. There are four general terminologies that we'll use when we consider controlling microbes. First is sterilization. That is the destruction of all microbial life. In disinfection, we destroy most microbial life, reducing contamination on inanimate surfaces. The key here is the inanimate surfaces. Antisepsis is destroying most microbial life on living surfaces. So the key being the living surfaces. Those are how we distinguish disinfection and antisepsis. Decontamination is the mechanical removal of most microbes from either animate or inanimate surfaces. So we can decontaminate the skin, or we could decontaminate a table. So this figure summarizes those four ideas. We've considered many of these modes before, but there's a group of physical microbial control agents, chemical agents, and then mechanical removal methods. And each of them could result in a variety of different types of microbial control. When we look at physical agents and consider heat and dry heat, in the incinerator or dry oven, we might end up with complete sterilization. Steam under pressure may also give us complete sterilization, but just boiling water, as we've considered already, not sterilization entirely, but definitely disinfection. When we consider mechanical removal methods, we're just dealing with decontamination. Again, under the chemical agents, we could have gases leading to sterilization or disinfection. We could have liquids. If those liquids are used on animals or living surface, it's antisepsis. If they're used on inanimate surfaces, we can either have disinfection, which is the correlation to antisepsis, or complete sterilization. Now, on an animate surface, like human skin, it's highly unlikely that we'd use a mechanism like sterilization. For sterilization, it would kill our own cells also. So the primary targets of microbial control are microorganisms that can cause infection or spoilage, or that are constantly present in the external environment. They fit into three general categories: those with highest resistance, those with moderate resistance, and those with the least resistance to microbial control agents. Those with the highest resistance are things like bacterial endospores and prions, and I bet that's no surprise. Now, prions are extremely resistant to heat and chemicals. Items that are suspected of being contaminated with prions should just simply be discarded. In this discussion, we're going to stick to non-prion sterilization, with bacterial endospores being the toughest or the most resistant microbes to our control agents. Those in moderate resistance are things like protozoan cysts, of which we've seen a few so far, some fungal sexual spores, naked viruses, and resistant vegetative bacteria. Now, some of the resistant vegetative bacteria are like Mycobacterium tuberculosis or Staphylococcus aureus or the Pseudomonas. In general, naked viruses are much more resistant than the enveloped forms. Hepatitis B and polioviruses are the most resistant of these enveloped form of viruses. Now, in the category of least resistance are most bacterial vegetative cells, fungal spores, as in the non-sexual kind, enveloped viruses, yeasts, and protozoan trophozoites. Those are the non-cystid forms. This is a comparative figure on the requirements for destruction of these various groups. Because bacterial endospores are so much harder to destroy, their destruction is the goal of sterilization. Any process that'll kill them will kill less resistant forms. So here we have the dosages of each of these different methods that will kill endospores. So we need 120 degrees Celsius of moist heat, 4,000 gamma rays, 1,200 milligrams per liter for sterilizing gas like ethylene oxide, or 3 hours in a sporicidal liquid. The vegetative forms require much less because they're less resistant. If we look over here, we'll see basically the summary that endospores are much more resistant. It takes 1.5 times as much heat, it takes 4 times as many gamma rays, it takes 1.7 times as much gas, and 18 times the amount of time in a sporicidal liquid. So endospores are much more resistant than our vegetative forms in general. So now let's take a look at the distinction between sterilization, disinfection, and antisepsis in a little more detail. There's a growing terminology that describes and defines measures to control microbes. Keep in mind, though, that the everyday usage of these terms is not always correct. For example, we might say sterilize or disinfect a patient's skin, when neither fits the technical definition. The following terminologies are used in antimicrobial control. Sterilization is the process that removes all viable microorganisms, including viruses and bacterial endospores. So it's the highest degree of microbial control. The material following sterilization is said to be sterile. This is usually reserved for inanimate objects for obvious reasons. To sterilize our skin would probably mean that we were killing some skin cells also. It can be a pretty harsh process. Mostly sterilization is going to be performed with heat, although sometimes we'll see chemicals called sterilants used in the process of sterilization. Again, the process of sterilization is for inanimate objects. Consider that something cannot be slightly sterile or almost sterile. Sterilization is a term that needs to be used in the strictest sense. And again, sterilization of the human body would call for really harsh or impractical methods, and it probably would cause damage to our own cells. Now you might ask the question, why don't we just use sterilization for everything? Well, the human body situation, that one's pretty obvious. But you got to think of the cost and efficiency of sterilization. It's not always going to be necessary, and besides, sterilization doesn't necessarily mean clean. In many situations, sterilization is simply not necessary. For example, destruction of spores is not always necessary. Most human infections, after all, are not from spore-forming microbes. So often we'll use disinfection or antisepsis. In contrast to sterilization, we'll look at disinfection, which again was for use on inanimate objects, so the tabletop. Disinfection is the use of a physical process or a chemical agent or disinfectant to destroy vegetative pathogens. It does not destroy bacterial endospores, but again, that's not always necessary. Most infections for humans are not derived from endospores. Disinfection can also be used to remove toxins from surfaces and might involve something like a 5% bleach solution like you've been using for your labs, or boiling water, or iodine solution. Concentrations that are effective, though, can also be toxic to human and animal tissues, so it's something we need to really consider. How much of a disinfectant do we use? For example, if you used 5% bleach solution on your skin for a prolonged period of time, we might see some issues with that. Antisepsis is much more appropriate for use on human skin. This is the means for microbial control that we usually use on tissues, right? So animate objects or humans. Antiseptics are applied directly to exposed body surfaces and mucous membranes to destroy or inhibit vegetative pathogens. So the difference between antisepsis and disinfection is antisepsis is technically on animate surfaces or tissues. Sepsis is the actual growth of microorganisms in the blood and other tissues, and thus the word antisepsis makes sense as being to prevent the growth of microorganisms in blood and tissues. Asepsis is the practice that prevents the entry of infectious agents into sterile tissues. Aseptic techniques that we've been practicing in our lab and are commonly practiced in healthcare range from sterile methods that exclude all microbes to antisepsis. Antisepsis is the application of antiseptics. We might use iodine in skin preparations for surgery, or hydrogen peroxide in the process of a root canal, and then hand washing with germicidal soap to remove microbes prior to surgeries. The terms sterilization, disinfection, and so on refer to processes. You'll encounter other terms that describe the agents that are used in the process. These terms are things like bactericidal or bacteriostatic. So let's take a closer look at these terms that describe the agents used in the processes. Cide means to kill. So a bactericide is a chemical that destroys bacteria. Again, not the endospores. A fungicide destroys fungal cells. So it kills fungal spores, the hyphae, and yeasts. A virucide is a chemical that inactivates viruses. We say inactivates because we aren't really considering that viruses are technically alive. A sporicide can destroy bacterial endospores. Then we have the terms germicide and microbicide. These are chemical agents that kill microorganisms in general. Now, stasis and static means to stand still or prevent multiplication. So a bacteriostatic is different than a bactericide because a bacteriostatic will just hold a population constant. The same with a fungistatic agent. They simply hold a population at its present level and prevent further multiplication. Again, cide to kill, static to hold still. Often, antiseptics and drugs that are used to control microorganisms in the body are called microbistatic organisms. So as you probably understand at this point, a microbicidal compound would be highly toxic to human cells also. So antiseptics and drugs used on humans, microbistatic. Finally, let's consider decontamination. Sometimes sterilization, disinfection, or antisepsis are not necessary. We often only are interested in reducing the levels of microorganisms or reducing the microbial load. So decontamination is used when actual sterilization isn't needed, but we do need to decrease the risk of infection or spoilage. For example, we'll see decontamination in the food industry. Sanitization is any cleansing technique that mechanically removes microorganisms to reduce contamination to safe levels. Restaurants, dairies, and breweries practice sanitization to keep microbes to a minimum. We'll use air sanitization with ultraviolet lamps, and then dishwashing machines are used to sanitize dishes. A sanitizer is a compound such as soap or detergent that sanitizes. We'll see how those work here shortly. The term sanitary means that it may not be entirely free from microbes, as in sterilized, but it is safe for normal use. These would be your regular restaurant dishes and such. Now, a term that's specific to reducing the number of microbes on the human scale is degermation. So degermation can be accomplished with things like alcohol wipes. So why wouldn't we sterilize restaurant dishes? Well, again, the cost of sterilization is much greater, and it's overkill in most cases. Sanitization is usually enough. But consider this. Previously, I brought up the idea that sterilization doesn't necessarily mean clean. You can have an absolutely sterile fork that still has food on it. It's been through a sterilization process, so the food is sterile also. But it's not very appetizing to receive a fork with food on it at your table, even if it is sterile. Or the same idea with a glass that has lipstick on it. It could be sterile, but it doesn't mean that it's clean to our sorts of standards. So the bottom line here is sterilization is often not necessary. Sanitization is plenty in most situations. Now, you might be seeing a little bit of overlap here between the concepts of antisepsis and degermation. They clearly overlap. The degerming procedure can simultaneously be antiseptic and vice versa. So don't worry about the overlap there. So let's extend on those practical concerns in microbial control. We have to ask the question, does the application require sterilization, or is disinfection going to be adequate? Is the item to be reused or permanently discarded? If it's going to be reused, can it withstand heat, pressure, radiation, or chemicals? So which mechanism should we use? Is the control method suitable for any given application? And then will the agent be able to penetrate to the necessary extent to see the desired effect? And finally, is the method cost and labor efficient? And is it safe? So now that we've taken into consideration some of the terminology associated with microbial control, how do we actually define microbial death? Because it's a lot more difficult to see in a microscopic, single-celled or very few-celled organism. When we consider large multicellular organisms, it's pretty easy to see. We see that the nervous system becomes dysfunctional and everything shuts down, and we can see what death looks like. However, when we look at a cell, just the fact that it's become still doesn't necessarily mean that it's dead. So we define death as when various cell structures have become dysfunctional and the entire cell sustains irreversible damage. The way that we can really establish that this has happened is that the cell has lost its reproductive capabilities. So we've got irreversible damage to the cell's machinery. It's no longer able to grow or metabolize. The permanent loss of reproductive capability, even under optimal growth conditions, is the accepted microbiological definition of death. So there are several factors that can affect the death rate. Because many microbial agents target the cell's metabolic processes, active cells tend to die much more rapidly than older or inactively cells that are less metabolically active. The effectiveness of a particular agent is governed by several different factors. First of all, the number of microorganisms. The more microorganisms, the longer it's going to take to kill off the population. The nature of the microorganisms in the population. So how resistant are they? The temperature and pH of the environment. If it's optimal, then they'll be killed off less rapidly than if the temperature and pH were less optimal. The concentration of the agent involved. Naturally, the higher the concentration of the microbial control agent, the faster we're going to be able to remove the microbes. The mode of action of the agent. How does it actually affect its action on the microbes? And also the presence of solvents, interfering organic matter, and inhibitors. On the right side here, you can see a few graphs representing these factors affecting death rate. First of all, the longer the time, the greater the degree of microbial death we're going to see. Now, this is an exponential curve really, but it's graphed in a logarithmic function, so it looks like a straight line. Eventually, we reach a point to where the population of microbes is so few that we consider it sterilization. Next, let's consider the time for sterilization as it relates to microbial load. If there's a large load, as in lots of microbes, then we'll see that it takes a longer time to reach sterilization. However, a lower load would take less time to reach sterilization. Now, the nature of the microorganisms in the population. We already know that spores are more highly resistant, so it's going to take longer of exposure to this microbial control agent for spores to decline in population than it is for vegetative cells. This graph illustrates the difference between microbistatic and microbicidal. At this level, we can see that the microbes are being held by a microbistatic agent. Then we add a microbicidal agent, and the population declines quite quickly. So the next question is, how do these antimicrobial agents work? What is the mechanism of action for these antimicrobial agents? They're going to affect one or more cellular targets, and they fall into four general categories. Antimicrobial agents can affect the cell wall, the cell membrane, the proteins and nucleic acid synthesis, as well as protein function. Let's begin by looking at the effects on the cell wall. The cell wall is there, and it maintains the structural integrity of both bacterial and fungal cells. A cell deprived of a functioning cell wall will become fragile, lyse easily, and so damage can often imply its death. Several types of chemical agents block cell wall synthesis. Others digest the walls after synthesis or break down its surface. Once the cell becomes fragile, it can break apart. Things like detergents and alcohol can also disrupt the cell wall's integrity, especially in gram-negative bacteria. Next, cell membranes can be affected by the antimicrobial agent. As you'll recall, cell membranes provide a two-way system of transport across the membrane. If the cell membrane becomes destroyed, that means that the cell cannot prevent loss of vital molecules that it requires for survival, nor is it able to bar entry of damaging chemicals. So, all microorganisms have a cell membrane. Many viruses have a cell membrane also. Detergents or surfactants are things that disrupt that cell membrane. They're polar molecules. That is, they have both a hydrophobic and hydrophilic region. The surfactants will bind to the lipid bilayer and penetrate into the hydrophobic region of that bilayer. This penetration of surfactants causes the membrane to break apart a little bit. This causes spaces in the cell membrane and allows the passage of previously excluded molecules into or out of the cell. This is exactly the way that your detergent dish soaps work in order to reduce microbial load. The surfactants disrupt the cell membranes, they cause them to break apart. Once there are spaces like this in the cell membrane, then they're no longer selectively permeable, and the cell can die quite rapidly. The next level that antimicrobial agents can work on is the level of protein and nucleic acid synthesis. Any level can be affected. Replication could be affected, transcription and/or translation could be affected. I've already introduced the idea that many antibiotics are targeted at the transcription and translation mechanisms. And I've discussed that some antibiotics bind to the bacterial ribosomes in a way that prevents the formation of peptide bonds. Thus, you're inhibiting the manufacture of proteins. Without these proteins that are necessary for growth and metabolism, the bacteria can't multiply, and thus we'll consider them dead. In the same way, nucleic acids are necessary for normal cell functioning. There are agents that can impede the process or even change the genetic code, thus rendering the bacteria unable to reproduce. And as you'll recall, bacteria that are unable to reproduce are considered to be dead. Finally, there are mutagenic agents that can interrupt protein and nucleic acid synthesis, like gamma, UV, or X-ray radiation. These cause permanent inactivation of the DNA. Formaldehyde is a mutagenic chemical that also interferes with DNA and RNA function. So if we have no viable proteins and nucleic acid synthesis is disrupted, then the bacterial cell will naturally not be able to metabolize and not be able to reproduce, and so will be considered dead. The final way that these antimicrobial agents can work is in protein function. The native state of the protein is its normal 3D configuration. Many antimicrobial agents work by denaturing or changing the native state. Thus, the protein can no longer function. When we disrupt the native state, it's called denaturing. What happens here is that the bonds are broken in secondary and tertiary structures. You'll recall the alpha helices, beta pleated sheets, and then the initial folding of the protein. Let's say the protein's normal shape is this. When it becomes denatured, it forms a different shape. Or maybe it's been exposed to some metallic ions that attach to its active sites. Either way, the active sites are no longer available for the original substrates that the protein is supposed to pick up, and thus the protein has been rendered inactive. When a protein's become completely denatured, it'll actually form a straight chain form. Now, coagulation is another thing that can happen. Coagulation is something that often happens when a protein has become denatured. We see this when we fry an egg. The egg white becomes denatured. It's this moist heat that causes the egg white to be permanently put into this denatured state. Now, there will be a video associated with the online textbook animation that I would like for you to watch that'll illustrate how this coagulation process happens. It's fascinating. Also, alcohols, acids, and phenolics could cause coagulation in this way. So that brings us to the end of this section. Before we move on to the next section, though, let's have a quick concept check. What type of compound is isopropanol when a nurse uses it to wipe your arm before giving you an injection? You don't have to understand what isopropanol is, but the usage here should tell you what type of compound it is. Is it antibiotic, disinfectant, a mutagen, or an antiseptic? Well, the definition of an antiseptic is something that's used on a living surface. Our skin would be a living surface, so that's definitely an option to consider. A mutagen, it's not causing permanent damage, we can cross that off. A disinfectant is for an inanimate surface, so we can take that off the list. And as you already know, antibiotics are used to prevent microbial growth rather than to clean off the surface. Antiseptic is our correct answer in this case.